course, I'm excited about the gene editing. And so the CRISPR-based gene editing, which has recently been approved in the UK, in thalassemia first, and then in sickle cell disease. In sickle cell disease only 2 months ago. It's based on the concept of editing the BCL11A gene to boost the fetal hemoglobin production. So we are, of course, looking forward into expanding the accessibility of this treatment to those who are eligible, but eligibility remains a challenge to define who is eligible for this and who can access this. Then in terms of thalassemia, we know that Luspatercept is approved in many countries. It is licensed in the UK, but we don't have any access to it yet. It's a novel erythroid maturation agent that reduces the transfusion needs in beta thalassemia. It promotes late-stage erythroid maturation, leading to increased hemoglobin production and reduced ineffective erythropoiesis. So the long-term treatment analysis up to 6 years now showed a mean 20% reduction in transfusion burden in the transfusion-dependent thalassemia patients, and stabilization or improvement in the iron parameters, which could therefore be translated as potentially reduction of chelation requirements and iron overloaded related complications, but also chelation toxicity complications. But also it has been shown to increase the hemoglobin in the non-transfusion dependent thalassemia patients, and we have a lot of research to support that increasing the hemoglobin decreases morbidity and mortality in the non-transfusion dependent thalassemia patients. So this drug could not only decrease morbidity and mortality in these patients, but also it could potentially stop those who would become transfusion dependent later on in their life. And again, from the literature, we know that about 20% of those who are not transfusion dependent for the majority of their lives or starting their lives will then be rendered transfusion dependent. So it's a drug that I have seen from the clinical trial phases. So for years now, being applied to patients that I look after and that I have followed from the literature point of view, which definitely would have a benefit in a lot of patients, but accessibility at the moment, especially in the UK, remains a problem. And there are a lot of unknowns that we're still looking into in terms of clinical efficacy and other problems associated with it. Nevertheless, it is a welcoming novel treatment. And then we have pyruvate kinase agonists such as the Mitapivat. This is a PKR activator. It's an oral drug which enhances the ATP production in the red blood cells, therefore improves red blood cell survival and reduces hemolysis. So in beta thalassemia, it has shown promising results from the clinical trials in increasing the hemoglobin levels, particularly in the non-transfusion dependent thalassemia patients, but it may be beneficial in the transfusion dependent ones by reducing the transfusion requirements. And there is some data showing that it might be also beneficial in sickle cell disease by reducing sickling and hemolysis, but research is still ongoing. This morning at the 4th Translational Research Conference, so the European School of Hematology Conference, we heard about exciting anti-inflammatory, if you want, molecules in sickle cell disease, which are early on in the clinical development. But it was very interesting to hear how these molecules, these drugs, might tackle different steps in the inflammation activation cascade in sickle cell disease.